So here we go. The recording has started. Hi, everyone. Thanks very much for tuning in today. It is a pleasure to host today Aleksandra Walczak, which is now part of our series officially, which makes us very, very happy because we have been chasing her for a little while. And yeah, today she will be talking about the scales of viral host co-evolution, which is a super exciting topic. So Alexandra already mentioned in the beginning that she has a hard constraint with leaving on time. So we need to finish by 6:04. If you have any questions during the talk, then please ask anytime. There might be the possibility that we don't reach the end, which is okay as well. But like if there are any questions during the talk, please tune in right away. If you're feeling a bit shy because we do record our talks, then you can also post in the chat and I will read them out loud. So far the housekeeping items. Let's go to Alexandra, and I will first introduce her bio a little bit. So like it is very impressive when you go through her CV, what she has done and to what schools she went to. In 2002, she finished her master's in physics, which aligns very well with our other speakers because they've been predominantly physicists so far, which is quite funny. But from the very beginning, she started with biology as well, and her master's thesis was on incorporating the conformational changes of proteins in the titration calculations. Excuse me, please. And then afterwards, she did her PhD at the University of California in San Diego, where she worked on stochastic nature of genetic switches and networks. And then she stayed there for another year in order to finish up some projects as a graduate fellow at the KITP at the University of California in Santa Barbara. Excuse me, please. Yeah, I need to correct myself. And then afterwards, she finished her postdoc in Princeton University in 2010, where she worked on the information flow and gene regulatory networks. And then she moved to Paris, where since then she's been a permanent researcher at the ENS. Since 2012, she is also an HDR, and since 2016, she is a directrice de recherche, which means that she can also lead and govern PhD students. So whenever you are excited about her projects, you can contact her. And if you're interested in like PhD or postdoc positions, then definitely do contact her. And at the moment, her main interest is in understanding the link between function, development, and evolvability of living systems. And then without further ado, because we want to get as much as possible out of your presentation, I want to hand over the mic to you. Alexandra, the floor is yours. Thanks very much for being today with us.
All right, well, thank you very much for the invitation. Yeah, what I'm going to try and do today is present work we've been doing over the years with Thierry Marat at the ENS here, joined by many people locally and experimentalists elsewhere. But I'm mainly going to try to show you how to think in a more abstract way about viral host evolution. And to start off, let me just briefly sort of get some things down about the immune system, which is what I'll be talking about. So when I mean host, I'm really thinking about all of our immune systems that are made out of a set of B and T cells. And these are cells that we have in our body. They have receptors on the surface, and these receptors bind different molecules and perform a discrimination task that they're able to discriminate the molecule that is natural to our body, which we'll be calling self, and we'll be getting back to that, and a foreign molecule, which is probably of pathogenic origin. And so when they recognize the pathogen, they proliferate, meaning they make many copies of themselves. And cells that don't recognize anything, after some time, they die. So there's some sort of population dynamics going on here. And over the last couple of years, well, now a dozen years or so, we have access to the diversity of these different cells that provide us with immunity, which we call a repertoire. We can go in and we can sequence these cells. We can sequence the genes that code up for these receptors that they have, and we can get some insight into the diversity. And so why do we need diversity? Well, we need diversity because there's a lot of different pathogens out there. And while each one of these cells has many receptors on the surface, they can actually, each cell has only one type of receptor. So to protect us against the many different pathogens, we need to have different types of receptors which recognize different bugs. We need a diverse ensemble, so a diverse set of different cells that carry different receptors. This is what we call a repertoire. And so this set, oh, okay, there's a question in the chat already. Okay, that's from you. It's not a question. So basically, why are we talking about adaptation here? Well, because these cells come in different frequencies, and as they proliferate, these frequencies change. And this is how the system encodes memory of what it's seen before. So basically, through these changing frequencies, we have some ability to change in response to the changing environment, which is the pathogenic environment, and our immune system can adapt to best, well, what I'm going to hypothesize is best protect us. And so the relative frequency, or not the relative, but the frequencies of these immune cells basically are like a personalized medical record. If we knew which cells actually protect us against, say chicken pox, we could go in, sample them, and we could know whether this person had chicken pox or not in the past. So that's what I mean by that it's a personalized medical record. And so the one sort of challenge in the field is learning how to read it. But that's not what I'm going to talk about today. Today, I'm going to think about this problem in a more abstract way, sort of in line of adaptation. And I'm going to introduce a concept that comes from Alan Perelson and collaborators from the 80s, which is the concept of a phenotypic antigenic space, which is sometimes called shape space. So imagine a space where these receptors and pathogens live together. They live in the same space, and the closer they are in this space, the more likely an immune cell is to recognize a given pathogen. Okay, and so I'm drawing this space as a two-dimensional space, and then the name of the game is to cover the space with immune protection. Now, each receptor gives me immune protection exactly where it is, but it also gives me immune protection in some region around it, which I'm drawing here as the sphere. This is a well-known immune concept of cross-reactivity, meaning that a receptor doesn't just recognize, or a cell with that receptor doesn't recognize just one type of pathogen. It recognizes many different pathogens that can be similar or not in sequence space, but in this effective phenotypic space, we assume that they are close. The pathogens that are recognized are close to each other. And so again, the closer you are, the more likely you are to be recognized. And if you fall as a pathogen within a ball of a receptor, then you'll be recognized by it. So a pathogen here will be recognized by this cyan receptor, the one here by this red one, and a pathogen in the middle can be recognized by both. Okay, so then the name of the game is to cover this space. And that way we're protected against all possible pathogens. Is that clear, more or less? Okay. So now you could be asking yourself, why am I drawing this in 2D? The answer is honestly that paper is two-dimensional and it's easy to draw things. However, there are other reasons that are not terribly convincing, but for sort of, for fullness of exposition, I'll present them. So people measure the response of different immune cells against different viruses. So for example, imagine different influenza strains, and you take a bunch of immune cells and you figure out which ones recognize this virus. And then you can do low-dimensional scaling, and you see that this is original work from 2004, but people have redone it since then, that you end up with these clusters in two-dimensional space. So these kind of look like that. So maybe that gives you a warm and fuzzy feeling that this idea is not completely nuts. But You may also ask yourself, well, why did, why 1 to D? And here the answer really is because paper is two-dimensional. And, you can do it in different dimensions. And there's a big question about what is this dimensionality of this space that nobody really knows. But let's go with it. And I'm sort of going to work in this phenotypic space and try to sort of see how far we can get with understanding what the immune system should be doing to protect us against pathogens. So in a way, how should it adapt to the pathogenic environment? So we really have two scales here. There's the global scale, which I've already described, that we want to cover all of our space with receptors and with immune cells that recognize different viruses. So if you have influenza here, HCV here, some other virus here, you basically want to protect it against every virus. And in previous work, we asked what is the optimal coverage strategy? How should you distribute your receptors given a distribution of pathogens? And we found out that you should do it more or less proportionally, but giving a little bit less weight to pathogens that you see. Okay. But then there's a second scale, which is a scale that is specific to a given virus. So if I say zoom in on influenza, I know that influenza evolves. And again, these two-dimensional maps sort of track influenza evolution. And I can start asking, how should my immune system change in order to be best adapted to protect us against future strains of, say, influenza? So basically, is my immune system able to do that? And in order to be able to sort of prepare and adapt to future influenza strains, two things need to happen. First of all, evolution needs to be correlated in time, because if evolution is uncorrelated in time at any given time, if I'm just sampling, if every time point is random and uncorrelated, then I have no chance of predicting anything. Right? So again, what I'm trying to do is answering the question, how should I update the statistics of these receptors? So how should I change how many bolts of a different type I have to be best protected against future strains? And I'm going to ask this question on very long evolutionary timescales. I'm going to ask, how should I design update rules for the immune systems to achieve that? So I need the environment to be correlated, but it is correlated. We know that viruses evolve due to evolutionary processes, which are correlated in time. And the other thing that I need is that I need to have memory. Even if the environment is correlated, but I'm not able to remember what I saw, then I'm not going to be able to predict anything. I'm not going to be able to exploit these correlations. So luckily, the immune system has what's called a memory repertoire. That means that after this expansion, after the immune cell has recognized pathogens, these cells proliferate, meaning they make many copies of themselves. And after that, some subset of them gets set aside and given a special flag, which basically means now they're part of a memory repertoire. And the next time the same immune challenge comes across, so the same pathogen, these cells can respond more quickly. And they also have their own population dynamics, their own homeostasis. They keep the size separately from new cells that are constantly being produced. So basically, it's known that the immune system has a molecular way of keeping memory. And the question we want to ask is, how should we update this memory? All right, so we're going to do this at the level of the specific virus and ask what is the optimal update to be best prepared for the evolution of this virus. So here we are in our phenotypic antigenic space where we're going to work with. And as I said, antibodies is just a different word for immune cells, and antigens, which is a different word for viruses, live in the same space. And we want to be as well protected against possible future viral strains, future viral escapes, so future viral environments. So we want to come up with global rules that will update our immune system in such a way as to be best prepared for the future. Okay, so we have a virus. We have a current immune cell here with its red ball of cross-reactivity. If the virus is here, this immune cell will evolve, it will mutate. So these antibodies or B cells that make these antibodies have the ability to somatically mutate. So they change the receptor in such a way as to recognize the virus. So what will happen is it will actually mutate like this, but then the virus, feeling this immune pressure, will mutate. And it does so rather at the population level. For influenza, for HIV, it can happen in us, but for influenza, it's not that it happens in each one of us, but it happens at the global level. The virus, feeling the pressure of all our immune systems, will evolve and jump. If it jumps here, well, our immune system, and we get reinfected by the virus, our immune system can still recognize it. It'll fall within the ball. But if it jumps outside of the ball, then, it cannot. So then the question is, how should we update our immune system? Should we just track it like before? Or should we now diversify and do some bet hedging and produce many cells that where one of them will actually hit a virus? Or and if that's the case, how many of them should we produce, and within what radius, given that the virus jumped so far? So what is the diversification of our immune cells, given that the virus jumps this strength? And on top of that, we have another form of machinery, which is basal protection, meaning that we always have, first of all, our naive immune system, but also our innate immune system, basically other mechanisms for which we constantly have to pay, but they provide a small level of immunity. So they're not as good, but they're always there. So which one of these systems should we use, and in which case? So basically the question we'll be asking is, what is the best strategy to protect ourselves against future viruses, meaning how many new immune cells should we produce, and in what way? How far should we spread them? Okay, so hopefully this is now starting to look more as an adaptation problem. So given that I have this ever-changing environment, how should I adapt the system that's recognizing it such that on long time scale, I maximize my survival, meaning I minimize my cost of infections. Okay, so to put some math onto this, I'm going to define a cost function, which is basically my minimum cost of infection. So on one hand, I can be protected by this basal green background, or I can, which is a high cost, but I can also have a cost from memory, which is generally lower, but is more specialized. However, I have a metabolic cost when I produce these new cells. I have to pay a cost because I'm actually producing something. So this is the number of memories for which I pay a cost. Okay, so this is constant, and this is changing and depends on the frequency of the frequency of the immune cell, and this is this cross-reactivity radius that we already talked about. So basically, if I have an antibody, I sum of all the immune cells I have modulo the red balls, and that gives me the protection I have from memory. And I weigh that against this higher cost, and I take the minimum one. And my goal is to minimize this cost on long time evolutionary timescales. Okay, and I can grow these cells. And I think I've seen everybody. I've said everything. So what I'm going to do now is by minimizing this cost on evolutionary long timescales, I'm going to come up with update rules for how I should update my immune cells from the current state to the future state. So I'm just going to derive update rules. Now, this needs to be done numerically. So if we do this procedure numerically, we identify three regions as a function of how strong this background naive coverage is. So this green region. So basically how well I'm protected against the unknown without any assumptions about what's going on in the environment, and as a function of how fast the virus mutates, so how far it jumps. So if the virus jumps very far, or I have very high coverage, I have very good coverage against this basal coverage, then the optimal thing to do is basically nothing. So there's two reasons for that. If I have very good background coverage, it's like having very good insurance, and basically it covers me, and I'm fine being protected by this non-specific response. However, if the virus jumps very far, it probably jumps so far that I have no chance of predicting where it will end up. And in that case, there's nothing I can do. So that's a very hard regime. So I might as well not try, not pay this metabolic cost. However, if the virus doesn't jump that far, or if this naive coverage is quite low, then if the virus doesn't jump very far at all, then the best thing I can do is track it. So this is what this monoclonal response is, and that's basically the red ball following the virus all the time. However, if it starts jumping much further, then that won't work, because again, my chances of tracking it decrease, and then I should do this ricochet type solution where I produce many different solutions that to cover my bet my odds, and try and basically and hedge my bets and figure out where the virus will end up. So if we look at this in sort of as what does the solution look like? The infection cost is being showed in color, so red is a lot. This is my evolving virus. So this is the regime where I'm actually able to track the virus. However, in this regime, I'm not able to track the virus because it jumps so far. So I'm just trying to produce these solutions wherever I can. Okay? Is that more or less clear? This is the problem
Alexandra,
of a Zoom talk.
I have a quick
Yeah,
question. So like you mentioned for the plot that you have in the top right, that the x-axis is the variance, so like how far the virus
yeah.
jumps basically, and that space over the radius, so that how far we can protect
Yeah.
the shape space, right? So this entire analysis is very much reliant on the dimensionality reduction that we put in place in order to represent this two-dimensional space, right?
Well, so we're not actually doing the analysis in two-dimensional. I mean, these plots obviously are done in two-dimensional space. I'll get back to the dimensionality of the space. It does definitely depend on the dimensionality of the space. And so this is what I just popped up.
Mhm.
So we can figure out where these transitions happen. We can do a slightly simplified model where these transitions actually are straight lines, because we assume everything is, the transitions are much sharper. And then, so not surprisingly, the response going from this one ball tracking to this many ball response happens when the virus jumps on the order of magnitude of the size of the red ball,
Mhm.
right? So that's not surprising, and this and the transition to basically the part where it's not worth tracking in any way happens when the jump uh, is of the order of the naive protection to the memory cost. So basically when the memory cost stops being worth it. But as you see here, there's this dimensionality parameter. So it's basically like, what is the size of the jumping ball measured in these radiuses to the power, to the dimension of the space.
That makes sense. Though, like, is there a specific meaning to the radius of that shape space? What would the radius here represent?
Right. So that's a question about what is shape space.
Yeah.
And that's a very good question. So you're really asking, can I translate shape space? I mean, basically the radius of this cross reactivity is the radius within which you are protected, right?
Hmm.
And we're modeling it as a radius, and we're modeling it as honestly a Gaussian,
Hmm.
right? A high dimensional Gaussian. But it in reality, it can be much more spiky than that, right? There can be dimensions in this space where if I make a few mutations, I'm already off the cliff, and other dimensions where I can go on for a long time and not lose protection.
Okay.
And so these are all very good questions, right? And the reason you may be having a hard time imagining that is because it's super hard to think about this problem in higher than 3 dimensions, right?
Yeah,
But
yeah.
it could be the answer. And in the theory we've done, there's nothing limiting you to 2 or 3 dimensions.
Yeah, that absolutely makes sense. That makes sense. Okay. I think that are all my questions. Thanks very much.
Okay, let me go on and I'll get back to this dimension question for in a second. So, okay, so we get these sort of, into, let's say, we justify that there's different strategies in the different regimes. It so happens that these two different kind of cells actually, these two kind of solutions exist in the immune system. There's cells that are called plasma cells that are the best binders. But normal memory cells are usually intermediate binders um, for a given virus. And people have been wondering for a while why, as a result of this process where the B cells mutate and produce better binders for the virus that is stimulating them, why do we also see these intermediate binders and not just the winners? And this is maybe a reason because, in fact, the immune system may want to be preparing for the future unknown, and not just dealing with the current problem, because by the time it's produced these best binders, honestly, the virus has been cleared by past binders. But just to say that these two kinds of solutions are actually seen in the immune system, and there's names for them. And honestly, either you care about this or just to give you a feeling that these are not completely crazy solutions. Okay. So, to sort of tie this back into data a little bit, so let's now think about what happens in this monoclonal regime where we have an evolving virus. So it starts off within the cross-reactivity radius at T0, and the virus mutates. And as it mutates and remains in the ball, then the number of immune cells that recognize this virus increases, and it steadily increases until a time t star when the virus finally evolves out of the ball, and then the immune cells that recognized it here start to decrease because they're no longer being stimulated. Okay, so we can actually calculate that there's exponentially distributed residence time of the virus T star within this ball, just assuming that it does a random walk. And then we know that there's a power law in the distribution of these abundances that's distributed exponentially again with this T star. So if we put these two things together, we're going to end up in a power law in the sizes of the of the so, sorry of the immune cells, of the frequency of the immune cells, if we put it together for different viruses. And the way it goes is that the more variable the strains are, so basically the faster they change, the smaller clonotypes, the faster the power law should fall off. So the smaller viral jumps the longer tails, right? Because smaller viral jumps means that the virus doesn't change very fast. So the prediction of this is that if the virus is slow, then we're going to get longer tails. If the virus is fast, then we're going to get smaller tails and sharper tails. And we can calculate this exponent. And again, it depends on the dimensionality of the space. And we can try to compare this to power law distributions in the frequency of these different immune cells and the sizes of the different immune cells that we see in data. And so we get these power laws. We can calculate these exponents, which are around 2. And then the thing is, can we actually try to, so we know do we have an analytical expression for the exponent? The problem is we neither know the dimension of the space, nor how far the virus jumps. So, but we can do an estimate having a constraint on this exponent. We can do an estimate of the viral divergence as a function of antigenic space dimension. And we can see that basically for all antigenic space dimension, the jump size of the virus is less than 1. So that puts us in this regime that we're basically around on, we're close to, well, we're less than 1, but relatively close to 1. So we're over here. So we're right at the border between this monoclonal and polyclonal regime, meaning we should be seeing these two types of responses. And it also means that strains are not unlikely to escape immunity because these two values are of a similar order of magnitude. But it also means that being in this regime is independent of the dimension of the space. So whatever the dimension is, with more or less, the constraint is the same. Okay? So it doesn't really help us put a constraint on the dimensionality of the space, maybe other than, exclude 1, which we knew anyway. Okay? But you can see that it connects somewhat to data. All right. So then we can sort of continuing in this space. So far, we didn't really worry so much about the virus. We worried more about the immune system. But now let's sort of think a little more about viral evolution. And I'm going to show you some more data. So as I said, like, for example, influenza, it's known that it doesn't involve in us, but it evolves from the pressure of all of our immune system. So we're in this regime where influenza evolution is basically driven by the population of our by the immune systems of us as a population. And people have been tracking influenza since the 60s and sometimes, well, precisely since the 60s and sometimes even before. And we see for the major influenza strain, we see this linear trend in evolution. So the color here is the number of different mutations starting from here. And this is just a representation. And what you should be seeing here is that basically it doesn't branch out. Okay, so the influenza strain that dominates at a given time stems more or less from the influenza strain that dominated recently in the past. Now, it doesn't have to be like that. Influenza B did branch in the 70s, and we have 2 branches of influenza right now. So just to give you an idea that it doesn't have to be that way. But since both SARS-CoV-2 and influenza look like that, and this is consistent with this image of basically the virus being pushed by immune system, and it gives us a way of thinking about it really as a one-dimensional problem where we're seeing this push of the virus. That's what we're going to do now. We're going to go to an effective one dimension and ask, when is the evolutionary process stable? Basically, when is it easier for the virus? What are the conditions for the virus to survive? And what are the conditions for the mutation rate of the virus to be stable? That's what we mean by evolutionary stability. And so we're going to see whether we can get some estimate of that. Okay, So now I'm going to, instead of working in a two-dimensional antigenic space, I'm going to work in a one-dimensional antigenic space, uh, which is the same idea. I'll have a distribution of viruses in it, a viral density, and I'm going to have a distribution of immune system density that basically pushes the viral density. This will result in the viruses evolving, basically this viral density moving with a constant speed. And the way is, but honestly, you sort of see it from the picture. You have some density of viruses here and it's being pushed, so well, it'll move. The two things to remember is again that we have these cross-reactivity balls. So the fact that I have an immune system here doesn't mean that my protection ends right where it ends. It means that I have this additional envelope coming from the green balls, meaning that I don't have to have an immune system precisely somewhere to have protection because it extends with the help of this ball. Okay, so you can think about it as a buffer with like a cushion that also offers protection, and that translates on the virus side wherever there is immune protection or wherever there is immune coverage. That means that the virus can't grow as fast as it would want to. So it only starts growing at its maximum fitness when all of this goes to zero. Otherwise, its fitness, so its growth rate, is reduced proportionally to this immune coverage. Okay, that's the basic picture. So I can stop there. If somebody wants to see some math, this is the math that goes behind it. There's an equation for the evolution of the virus, which basically says it grows and it mutates. The immune system, you acquire immunity where you have the virus, because that's how immunity works. And there's a compensatory loss term. We assume that the host population size is constant, but that the viral population size is not. So viruses can go extinct, viruses can explode. Obviously, viruses need hosts to survive. And the fitness, as we said, is proportional to the susceptibility, which depends on cross-reactivity. But in short, this is just putting equations on the picture that I just described. Okay, so I described the picture in 1D, but it also holds in 2D. So just to give you an idea of what it looks like in 2D to relate to what we were talking about before, this is a virus evolving in 2D. And as it evolves in its wake, it leaves this wake of immune protection. Okay. And the thing is that because of these immune systems, the effective speed of the weight is much larger than it would be from just mutations. It's really being pushed. Okay. The other thing that can happen, we can calculate, is how often it'll change direction. As you can see here, it changes direction. So there's some persistence time scale where it'll go in a straight manner, and we can calculate how often splits happen. I don't want to go into the details of these calculations. What I really want to say is that there's a order of time scales. So basically there's a smallest time scale, which is the mutation time scale of the virus, which is slower than the time scale on which the virus escapes the immune system, which is smaller than the time scale on which it changes direction, which is yet slower than the very, very large time scale, so rare event of splitting. But whether we can predict where the virus will be, so depends on basically whether it's turned and how far it's mutated. So it's somewhere in between these two timescales, depending on, again, the dimension of the space. And given that right now we can predict the flu on the timescales of 8 months a year, that puts a bound on, so basically putting in of the order of a year here, we see that this dimension is pretty high dimensional, by which I mean 6 or 8 dimension, right? If you plug it in. And I don't mean 100 dimensions, but basically it shows us that it is not 2 dimensions. If I plug 2 in here, I would get, well, actually, that's maybe not a good example. But basically, this dimension is higher than 3 dimensions.
Right.
Yeah,
Can I ask you again, when we're talking about the dimensions again in the state space, are we still talking about the same state space as
yeah,
before?
yeah,
This is still the same state
yeah,
space as before in the
yeah,
for the immune system.
yeah.
So here, how you have modeled this, that one of the underlying assumptions here is that you can move into any direction in your state space at any time point.
Mm-hmm.
And it is continuous, so there are no jumps anywhere.
Yeah, so in what we've done here, there are no jumps. We're now working out the version with jumps.
Oh, okay, okay, then I'm waiting.
Well, I won't give you the answer because this is a student who just started last year. But yeah, it's an interesting question what jumps will do. So because if you think about it, if I now jump here, then so the first thing that needs to happen is I need to overcome genetic drift. I need to survive as the new virus. But providing I survive, I mean, the most likely thing is I won't. But if I do survive, then I'm basically building an alternative population here. And then either this one catches up with me and they merge, or I just, this one will die and that'll be the new wave. And so describing all of this, and actually the jump scenario is a very relevant scenario because that's what happens with flu and SARS-CoV-2 of reappearing from different reservoirs and so on.
Yeah,
So I think to actually describe the data that I showed initially, you need to include the jumps.
okay,
But so far we don't have the jumps.
fair enough. Thanks very much.
All right. So the question I'm going to try to answer here, though, is this, what is the mutation rate that basically makes the virus coexist with the immune system and means that it cannot be overtaken by with viruses with other mutation rates. So what's the evolutionary stable mutation rate? Okay, but to do that, I need to look at 2 regimes. The first regime will be a regime of small cross-reactivity. So basically, when my green balls now are small enough that the green coverage line hugs very closely to where the immune system is, that means that very close out, if I don't have immune protection, the virus can grow as fast as possible and it has a constant fitness because it's growing at its maximal rate, basically the R0 capacity. And in this limit, the problem maps onto a Fisher KPP wave, which has been basically solved. It's basically a moving wave with a velocity given by the mutation rate and R0. I mean, this regime is very simple. Basically, if you have a susceptible individual and it meets an infected one, 2 will become infected, and the number of infected individuals increases in this way. The other regime is a little bit harder. It's the regime of large cross-reactivity, meaning even out here, when I don't have immune coverage, the virus still has a hard time growing because there's the cross-reactivity. So basically, every mutation it may take, it'll probably still fall into some ball of the immune system. So it's very hard for it to escape the immune system. So what happens here? Well, the virus tries, it mutates. Whoops. As it mutates, the immune system tries to push harder and it catches up with it. So as it mutates, it manages to grow, but the immune system quickly catches up and decreases the population of the virus. And so again, then the virus starts to grow. And there's a steady state here, which is given by how fast the virus moves times the selection pressure, which determines the effective size of the viral population in terms of the host. And you can work this all out and you can get a complicated expression for the speed of the wave. But the thing to remember is this is a very, very hard regime. And basically, the virus that manages to jump and survive has a huge advantage. So we can put these two regimes together, so we can figure out the wave speed in the two regimes, but we can also put them together and we can calculate the wave speed everywhere. And basically, there's a dimensionless parameter that controls it, which is the ratio of the virus growth to escape. And so, but just to say that there are these two regimes, which is really an easy regime for the virus and a much harder regime for the virus, because the most likely thing that can happen is that the virus will die. So the question we're going to try to ask is, what is the best mutation rate for the virus to have? So you may think that the virus should just mutate as fast as possible. Because, if it mutates fast, it runs away fast. But there's a cost because most mutations are bad for the virus. Basically, this is called a mutational load, but most mutations are deleterious, meaning they'll actually reduce the growth rate of the virus. So there's some trade-off between mutating very fast and avoiding all these bad mutations. So we're not looking here for the optimal mutation rate, but for the one that's evolutionary stable, meaning that any mutant that has a different mutation rate will not be able to displace the virus we have right now with its own mutation rate. So if you want it in math, basically any change to a mutation rate will result in a slower growth rate for the virus. Okay? So in that sense, we want to find the right mutation rate. And we can do that. And if we do that, then we find that the solution depends on the regime you're in. So in the small cross-reactivity regime, again, the easy one, the virus can only basically the thing for the best thing to do is to move as fast as possible. So if you're a mutant, if you're the resident virus, you want to move faster than any mutant. So from the immune system. However, in the large cross-reactivity regime, that's not going to work because moving fast will probably, so mutating as much as you can, will probably just lead to a lot of death. So what you want to do is you want to, every time you actually manage to escape, you want to capitalize on that escape. So once you survive, we've mutated and survived, you need to grow as fast as possible. So you need to maximize your reproductive ratio. So these are very two different strategies. And so basically the reason that you have to, it's not that this is generally a good idea, but here you're in a regime where you really don't have a choice and there's not much more you can do, that every time you get lucky, you use that as much as you can. Whereas here, you can just run away. All right. So given all that, the question now is, where is the current flu? Is it in a regime where it should be running as quickly away as possible? Or is it in a regime that that basically it should be exploiting any good mutation it finds? So if we were in a room, I'd ask you to vote, but that's not gonna work. But basically, okay, what we know is that the flu has an R0 parameter of about 1.8. Remember, 1 is that it is what's needed for keeping alive. And we have some estimates of cross-reactivity parameters plus our evolutionary stability condition and our speed of await, and that can give us an estimate of a mutation rate. And if we put all that together, we find that the flu is roughly here. So it's not in the impossible regime, but it's pretty close to the hard to escape regime of large cross-reactivity and always feeling the immune system basically breathing down its neck. And the reason we could have predicted that without basically doing all this analysis is because this R0 is not huge. Okay, it's still pretty close to 1, meaning that it's still feeling the pressure of the immune system on its back. But sort of what I guess I try to show you is mainly that doing these kinds of analysis, we can get some estimate of how the virus escapes and how the immune system tries to adapt to an escaping virus. So maybe I should stop here. I had a third part. I can just sort of advertise it. It doesn't really matter, but there's a missing element that I haven't talked about in terms of this effective space, where I said we want to cover it, and that's true, but there's forbidden regions in it, which correspond to your own proteins. So you don't want to recognize your own proteins. So the question is, should you cover space in such a way as not to hit your own proteins? So there's an interesting biological training process, but basically we went in and we tried to find signatures of these immune cells that have been eliminated because otherwise they would recognize these the self proteins. But in short, while we find some signatures, we don't find very strong signatures. So that's what the third part is about. But I think I can stop here in the interest of time and take questions.
Perfect. Thank you so much for your brilliant presentation. That was really, really interesting. First, to the audience, are there any questions? Anything that you would like to know? Marco, please go ahead.
Yeah, thanks for the for the talk. I was interested by the fact that, for example, for the influenza, you said that it's very unlikely to branch. And I find this really a bit bizarre because if you have that the evolution of the virus is due to the pressure from the immune system and you have a large population, everybody's infected, right? Then you would expect that the evolution of the viruses across different parts of the population, for example, could take different directions, right, in this high dimensional space. And I would say that that would be equivalent to branching. And so it's a bit strange.
Okay, so
that
maybe I can stop you. I mean, so
you understand what I'm trying to
yeah, I
get
understand
to,
what you're trying to say, and I think that the short answer is there's a lot of complexity, right? I mean, your question is really a question about the data, and the short answer is this is what the data is, but I think it helps understanding what the data actually is. So when it doesn't branch, we're really tracking the same strain, meaning that these are if we're looking at the virus of influenza virus now, then we're looking at its direct ancestors. But I think a lot of your intuition comes from the idea of niches, that you can start forming niches. And then it's true that I think different strains may feel more comfortable in different niches. Okay. And really, our global influenza, there's a lot of different influenza strains. There's a lot of different subtypes of influenza circulating at any given time. And what happens in reality, and this is why the jumps are interesting to model also, is that some of them go into animal reservoirs, which I think is what you're thinking about as a niche. They stay there and then they come back. Is that helping?
Maybe.
I'm just
Maybe.
basically I'm telling you that the problem is way more complicated.
Yeah,
But
of
yeah,
course.
it's a good question about, what do okay, this is something we've actually worked on, and I'm just having trouble summarizing it in a minute. But we did ask the question about how having a niche structure influences survival.
Mhm.
And we didn't look at it as a function of the dimension of the space, but we looked at it as a function of the topology of the niches.
Mhm.
And so, for example, having linearly, so you increase viral survival if you're in a regime where the virus jumps between niches. So first of all, having niches can improve viral survival, and the best way for the virus to do it is if it has a rate of jumping between niches that is fast enough, but not too fast, in the sense that when it jumps, it needs to start to occupy and diversify within a new niche. So it actually needs to generate heterogeneity such that then the next jump, when it jumps again, it jumps with a mutated strain than the one that arrived. Okay. And then if you understand that, then the fact that a linear topology is the best thing for viral survival becomes kind of clear, as opposed to, say, a star-like topology.
Okay, thanks.
Yes,
Georgie?
please.
Oh, yeah, thank you.
Somebody else?
Thanks.
Sorry.
Oh, maybe I jumped in. Is it okay? Is it my turn?
Yeah, absolutely.
Okay.
Okay.
Okay, thank you so much, Aleksandra. Beautiful talk as always. I have a question that you may have already answered, which is that what makes different viruses place themselves in different positions along the line between easy to escape and hard to escape? You made you gave the calculation for influenza, but I guess this depends on both the on the virus and the host, no? And there is a feedback there. I mean, there's a mutational cause, then there's the cross-reactivity. This changes
So
from
the
virus to virus.
cross-reactivity depends on the cost. There's the
Okay.
mutation rate, there's the R0, which also, I guess, depends on the host, right? How so it depends on, let me see if I can find you all the probably can't in this presentation on everything that it depends on, but it's basically the parameters that are more or less here. And that, I mean, the parameters in this problem are really capital R0 and small r0, the mutation rate, and virus population size and host population size.
And I imagine there's a large variability then among viruses. You gave the example of influenza, but
for the virus, I mean, when we did this, we basically varied the virus population size, and then we had some. I mean, to explain, you have two holds. One is an equation of evolutionary stability, which is just an equation that connects these parameters, and you have the speed of the wave, which is another equation. Okay, and then so both of these equations you can plot as a function of the mutation rate. They give you lines that if you say, I know what the cross-reactivity is, that puts me on a point on the first line that then translates into a point on the second line, and that translates into what the mutation rate should be. That's how the estimation works, and there's different lines depending on different host population sizes, but honestly, within this range, this doesn't change the estimates that much. Okay, so now if you ask for different viruses, I'd say the bigger question is, are you actually in an evolutionary stable regime? And I think with the flu, we're fairly confident we are. With SARS-CoV-2, I think we're getting there, but we're not there. Then there's different viruses like measles that just don't evolve for kind of biochemical reasons, so the question doesn't make sense. So it is all over, but unfortunately, due to our extremely biased sampling, that we only care about influenza and SARS-CoV-2 and sometimes HIV, we don't have that much data about what are called rare viruses, but that are actually not rare.
No, thank you. No, it's still very interesting, this view. Yeah, thank you so much.
Perfect. Well, thanks everyone. It is exactly 6 o'clock. Aleksandra, thank you so much for being with us today. If there's any other question, then Aleksandra needs to leave. She has already mentioned to me, but like if anyone in the audience has a question, you can email us and we are happy to forward them also to Aleksandra. Thank you so much for your wonderful talk again, beautiful work, and then I hope to see you very soon again.
Thank you, and thank you so much for having me.
Of course.
Bye,
Thank you very much.
everybody.
Bye-bye.
Thank you.
Bye. Bye-bye. Thank you.